Hello lovelies, in this video we're going to be looking at food tests for your GCSE biology. So this is one of the required practicals. It's one of the smaller required practicals, and then in the exam what they can do is give you some data, so we're going to go through how you analyse that, or give you kind of like a mystery component where they expect you to try and work it out what it is based on a series of tests.
Tests. Before testing any food, the sample needs to be crushed or ground up. You can do this in a blender or using a pestle and water. Then we add water to it. Then you get kind of a solution where you've got your dissolved food in the water, and we can place this in a test tube, and each test tube can then be tested using a different reagent. For this practical, so for any of these food tests, we should be wearing goggles because all of the reagents that we're going to use are irritants or harmful, so if they get in our eyes, they could damage them. Equally, if they get on your skin, then you will need to wash your hands straight away to get it off, and also obviously clear up any spills. Testing for starch. Starch is a long carbohydrate chain made of sugars, so we're testing for starch, we're testing for a carbohydrate. So we can use iodine to test for the presence of starch in food. The iodine solution, or potassium iodide, is yellow to start off with, and it will go blue-black in the presence of starch. Be careful, make sure if you're talking about how the positive result looks for iodine, you need to say blue/black or black, but you cannot just say blue in the exam. And remember, we say it's a positive result when there is a colour change that happens when the food or the molecule that you're looking for is present. Testing for glucose. So we use Benedict's reagent to test for the presence of glucose, so that's small molecule sugars in substances. It starts off as a bright blue solution, and it only works when we heat it up. And we have to say we heat it to above 80 degrees or we boil it.
You can see it has the brilliant blue colour there, and that comes from the copper sulfate. We need to add our Benedict solution to our sugar solution, equal volumes of sugar solution and Benedict solution. And then once they are mixed, we can see the colour of them there, the brilliant blue colour. Once they are mixed together, we can heat them. Now this does take quite a long time to heat. It does need to be quite a vigorous heat, and you need to be able to do this safely in a lab. But already you can see the colour change going on there as it goes from blue through green, yellow, and then to a dark kind of brick red colour. And this colour, this brick red colour, is a positive result.
So you can see in the picture here that we get that brick red, that dark red colour, and that's the positive result. Obviously, there's a range of colour changes that happen here with the Benedicts. So from blue, anything other than blue shows that there is glucose present. A green shows a bit of glucose, yellow shows a bit more glucose, orange shows a bit more glucose, and red shows a lot of glucose. So we have a range of colours to represent the fact there can be different amounts or concentrations of glucose present. Testing for proteins. We use the PhET reagent to test for proteins, which starts off as a pale blue solution. As with all the others, we're going to add the same volume of PhET to the food solution, and we're going to give it a mix. So although this is a blue solution and it begins with a B, it's not like Benedict's in that it is a reaction that happens at room temperature, so we do not need to heat it, you just need to give it a mix, and it should turn purple if protein is present. So a positive result for PhET solution is going from blue to purple. Testing for lipids. So we add ethanol to our food solution and we need to shake it. Then we add water. The positive result here is not a colour change, but we do see a white cloudy layer, and this is the emulsion. You can say that the emulsion or a white emulsion appears. You need to say white and emulsion. You can say cloudy, but you cannot just say cloudy on its own as an answer.
Ouch! This is why in some videos I explain scratches.